Again, thanks for the invitation. So my name is Zhe. I'm a PhD student working with Professor Stefano Sacanna here at NYU. So today I want to share with you guys how we can engineer the abiotic materials to create living cell mimics in the lab, especially on their ability to do the transmembrane transport. So the title of today will be the active colloidal microcapsules. But before I start, I'd like to give a brief introduction about who we are. So we are a group of colloidal designers working under the leadership of Professor Stefano Sacanna. The main goal in the lab is to create colloidal building blocks from the bottom-up approach, which means they are monodisperse and scalable. So we build up our own periodic table of colloids that can be used as the analog for the molecular species. There are two main directions in the group. The first is to do the self-assembly. We typically engineer the surface of particles so that they have a self-interaction. They can self-assemble into microscopic objects under the thermal fluctuation. One example here is our recent work published on Nature, where we engineer the surface of common colloids you can find in the market. We use the combination of electrostatics and polymer repulsion to self-assemble them into ionic crystals. The other direction in the group is to study active matter. These are the colloidal objects that not only obey the Brownian motion, but also respond to the external stimuli or energy input so that they can do more functional work. One example here is we can make these colloidal swimmers. Once you shine the light, they self-assemble into a colloidal gear that spins and can do mechanical work. So back to the topic of today, why are we interested in a colloidal membrane? So if you look at the evolution of life, one of the most striking structures the nature ever evolved is the colloidal membrane, or a membrane on the colloidal scale. And this is called a protocell. A protocell separates itself from the outside environment so that they can break the thermodynamic equilibrium and start to accumulate what they love and to expel what they hate. After which, there's the long journey of differentiation and evolution towards more sophisticated organisms until become our human beings. But interestingly, now we are going back to study how the original life forms. So in order to recreate the living protocell or artificial cell, there are two main criteria. The first, you need to have a membrane. So in nature, the life utilizes the bilayer of lipid that's flexible and can do the self-division in order to separate themselves from the outside environment. The other ingredient is you need to have active matter that can guide the membrane to do the functional work. So in nature, they use the combination of DNA, RNA, which guides the synthesis of many macromolecular machines that can do the work. So if you zoom in onto the lipid layer of biological membrane, it's a very complex system. So the nature utilizes the full toolbox of different micromolecules, including the lipid, protein, polysaccharides, to build up this membrane with many different channels on top. So they can realize both the passive transport and active transport to accumulate the cargo. So synthetic biologists and chemists always want to replicate this by sorting out the minimal ingredients you need to self-assemble the same structure. But you know, the nature took thousands of years to evolve the strategy. It's highly unsuccessful. It's more like trying to find the needle in the haystack. Colloidal scientists want to target this problem from another perspective, that is, we study the structure-function relationship. The idea is, if you can mimic the structure using even abiotic materials, you should be able to achieve similar functions. Some success has been made in the field. One is to use this generous particle to make active Pickering emulsions. So Pickering emulsion is just emulsion droplets stabilized by particles. These active Pickering emulsifiers can respond to external stimuli like electricity, light, or even magnetism, so that you can artificially open up an aperture on the surface and to do some transmembrane transport behaviors. The other example is similar, where they utilize the magnetic particles to open up aperture to do the artificial phagocytosis. But both methods are limited in the way that you can only produce polydispersed droplets. Droplets are pretty big. And in order to manipulate the droplets, you can only do that one by one. There's no global stimuli you can use to do the functional work for all of the droplets. So as a result, in this talk, I want to show you how we can make colloidal microcapsules as the colloidal membrane to do the transport work from a bottom-up approach. So the first part, I will talk about the self-inflation mechanism to transform common colloidal oil you can make in the lab into a bulk scale colloidal microcapsules. We can also add an extra layer of complexity to build a hole on top of the membrane so that they can be used to study the particle level transport. In the second part, I'm going to talk about the functions we can get out of this microcapsule. The first is the simple case, which is the passive transport. I'm going to briefly talk about how we can measure and study the transport on the molecular scale and the particle level scale. And finally, I'm going to talk about the active transport, which is to utilize the power of active colloids to transform the microcapsule into active cell mimics. So without further delay, let's start from how to make the capsules. So as a colloidal scientist, I'm going to use one of the most popular materials in the colloidal community. It's called TPM. So TPM is the organosilane molecule like this. It's very simple and cheap. When in the monomer phase, they are immiscible with water and they are just transparent oil. However, if you expose them to some weak basic solution like ammonia, this monomer will start a process called spontaneous emulsification. Basically, the methoxy groups will start to hydrolyze and condense together into this siloxane matrix. This forms the spontaneous emulsification of small droplets inside the vial. As you can see, the vial becomes turbid, and if you take a sample under the microscope, you get very uniform droplets underneath. There are two processes happening together. So firstly, the droplets will keep growing bigger and bigger over time as they incorporate more and more monomer species. The other one is the cross-linking inside the matrix also changes over time, as can be indicated by their vacuum behavior on the SEM stage. It's behave more like a solid particle over time. The special part of TPM is they also have this double bond on the side, which means you can always polymerize them in situ to fix whatever structure you observe in the liquid phase. And you can essentially keep these particles stable in solutions for years without any problem. So because of this easy preparation of the droplets and this facile transformation between different properties, TPM is very popular. And just to show some of the work TPM can be used, one is TPM can be used to make particles ranging from nano size to micron size, all uniformly and in the bulk scale, which is highly economic. No other oil droplets can compare with this. TPM also can change the consistency from liquid to solid, as I said, by polymerization. You can also calcinate it further to get a pure silica sphere. And all of the structures will be, they will keep the same shape you observe in the liquid phase. More recently, we also utilize the power of polymer absorption. You can graph the TPM droplets with the corona of steric polymer that introduce more interesting interactions to guide the self-assembly. We can also use shorter chronic polymers that mix with the TPM to form kind of phase transition. And you can get either microgel or anisotropic dendritic particles, which are all very hard to make for a liquid droplet. Although people have studied TPM for a long time, the two processes I mentioned before are usually ignored. That is the growth and cross-linking. These two processes are decoupled in terms of their time scale. If you monitor that, you will find the growth of the droplets usually saturates after hours. They reaches the maximal size. However, the cross-linking inside will keep changing even over days. This gives you the ability to recover droplets with seemingly the same size, but with very different characteristics inside. So when the droplet is very young, the main structure inside droplets is linear, which means they are very weak. And you can add strong base like NaOH to further dissociate the matrix, and the droplets will just dissolve. On the other end of the scale, if you keep cross-linking too long, all the structures are cross-linked into the siloxane matrix, and they will be resistant to any kind of attack you have, like NaOH, they will remain intact. There is the sweet point in between, where you do have this three-dimensional matrix to support the droplet, but they also have some dangling chains on the side that can be hydrolyzed and dissolved. When this is the case, we start to observe the self-inflation of our TPM droplets. So in this video, I'm going to take a lot of TPM droplets uniform, and I add NaOH. And all of a sudden, you start to see the water pockets formed inside that further coalesce into a single cavity. This is due to the reason that the side chains that are hydrolyzed are trapped inside, which causes the osmotic pressure imbalance. In order to support that, they will absorb water in order to blow up the cavity. The whole process happened bottom up, and there's no bias in terms of the size of the initial droplet. So we can successfully transform a whole batch of TPM liquid into a whole batch of TPM microcapsules. So as I said, there are two key parameters. One is the aging or cross-linking of the TPM. The other is the base concentration we use as the attacking agent to break the chains. So if you look at the phase diagram we built, there's a very wide window in between, which is the intermediate aging, and with suitable amount of base, the TPM droplets will inflate. And if you polymerize them, you can essentially get highly monodispersed microcapsules that just self-crystallize by themselves because they are so monodispersed. And we can even check the dimension under the SEM, and you can see such a cavity formed. So the self-inflation effect is also common in nature, like the pufferfish will engulf water when they are stimulated, and they reach the final size by balancing the elastic pressure from the skin and the water pressure they engulf. In our system, there's the simple balance of pressure to determine their final size. We have the surface tension from the liquid membrane that's trying to compress the capsule, which is balanced by the osmotic pressure induced by the low molecular chains that's trapped inside. However, you can also add something outside trying to shrink the capsule. Anything that's semi-permeable through the membrane can be trapped outside, and they will cause external osmotic pressure that also exerts the compressing force onto the microcapsule. From the experimental results, we found that NaCl cannot penetrate, and we can use this to selectively change the size of the microcapsule. When we add some salt to the solution, the microcapsule will start to shrink as predicted by our simple equation. And it's such a nice trend. As you can see, we can use the same batch of capsule, but we can get all different geometric design you want. Some are smaller but thicker, others are bigger but thinner. This is very important after you polymerize the capsules, because unlike a liquid capsule that will shrink upon the addition of external pressure, solid particles will typically buckle upon a critical pressure. This is called a failure. So depending on the geometry of your capsule and the materials of your capsule, they can buckle in various ways. This is the topic mechanical engineers are very interested in. And in our system, we observe two kinds of buckling. When the capsule is very large, very thin shell, you get something like a paper ball, crumpled paper ball on top. However, if you increase the shell thickness a little bit, they all buckle uniformly into a hemisphere. And because our capsules are very uniform, this buckling behavior is sharp, which means we can use this non-invasive agent, the NaCl, to trigger the buckling of the capsule and to release anything trapped inside. This tends to be a very good platform to be used as drug delivery, which we will investigate further in the future. Just to quickly summarize the first part, we introduced the self-inflation mechanism to transform the TPM oil droplets into the TPM microcapsules from the bottom-up approach. So when you build it up upon the initial spontaneous emulsification, you get a whole tunability in terms of the final capsule size and the shell thickness that can satisfy any kind of experimental design you need. However, there's no way for us to study a particle level transport with the original microcapsules because it's the closed membrane. So in order to overcome that, we are going to template a uniform hole on top of the membrane. So throughout the years, I developed various strategies to do so. One is called inside out, which is typically an extension of the self-inflation. So we start from the hybrid emulsion in this case, which is the TPM emulsion templated with the sacrificial seed. We use polystyrene here, which can be similarly inflated, polymerized, and dissolved to get this microcapsule with the monohole on each of the membrane. Sometimes we also want to get many holes on the surface. We have studied the outside-in approach to produce colloidal some that give you this porous membrane on top. I can even combine these two to produce the microcapsule with holes on only half of the sphere. Due to the limit of today's time, I will only introduce the inside-out approach, which gives us the monohole structure. So TPM has been studied for a long time. One reason is they can form the monodisperse droplets by themselves. The other reason is they essentially grows on any kind of colloidal seed you can imagine. This is called the heterogeneous nucleation. If I start the nucleation with presence of many polystyrene seed, especially negative, these TPM droplets will template on this seed and to form this hybrid emulsion. And because of the charge of the TPM is negative, polystyrene is negative, the wetting will cause the seeds to penetrate through the membrane once you inflate them. So if I polymerize this structure and dissolve the polystyrene, I will recover a mono hole. And this templating is purely designed by this kind of physical wetting. So all the holes will be the same as long as you use the same material and the same size of particles. This strategy is very universal. I can use this to template all different sizes of hole from nanometer to even microns using the same strategy. And you can also use other shape of holes. For example, I can produce inorganic particles that is anisotropic. Like I can use these magnetic hematite particles, which is alpha arrow oxide, and I can make a cubic hole or even a line-shaped hole on top of that. So combined with all other strategies I designed, I'm able to build up a library of microcapsules with all different shape and the distribution of the holes that I can further utilize to study the physical transport. So after getting these microcapsules, we are ready to study the transport behaviors. Let's first start from the passive transport. When we talk about the passive transport, either on the molecular level or on the particle level, what we are talking about is the diffusion coefficient. In the theoretical setup and the perfect condition, this should be inversely related to the size of your object. But we know that molecular species are very small, they move super fast, so it's very hard to visualize them directly. So we usually rely on indirect observations to induce whether they penetrate or not. Like salt can be used to buckle the solid capsule or shrink the liquid capsule, which tells you that the salt cannot penetrate. But we do need some quantitative solid evidence to support that. So in this chapter, I'm going to give a brief introduction about NMR diffusometry that you can use to give you the idea about whether they diffuse inside or not. Even on the particle level, like colloidal particles, you can see them, but the diffusion is still very complicated. Nothing is as clean as the ground emotion model, where they can just diffuse with no constraint. You can predict the time scale for them to go over a certain distance. As long as you have a constraint around them, this is called entropic bottling effect, and the particle needs much longer time in order to reach the hole and exit. And this is the very interesting phenomenon in the physics field because it's the very powerful inherent strategy to trap things. So let's start from the molecular transport. So molecular transport in the colloidal system is nothing like driving a car on the street road. It's more like navigating inside these interconnected labyrinths of channels. So engineers are very good at giving up some metrics trying to characterize the system, like tutorosity or porosity or even simple surface volume ratio. These are very useful when you are doing a simulation, but even the simple surface volume ratio is very hard to measure in the experimental setup. So NMR diffusometry comes into our hand as the very easy measurement. In very simple words, when you apply a rotational magnetic field, all of the electrons will spin with the same frequency over a certain distance. This will give you a very strong signal. However, I can add a gradient of extra signal that accelerates parts of the spins and decelerates the rest of them. If I wait for a certain time and if there's no diffusion, I can reverse the signal to recover the original strong intensity. But this is not the case if there's diffusion of molecules happening in between these two gradients. In that case, some of the spins will move out of phase so that when you apply the reverse gradient, they can no longer recover 100% of the coherence you get to start with. Because of this, we will get attenuation of the intensity over time if the spins or molecules are moving due to the diffusion. So this is the very simple exponential decay equation studied extensively by the NMR experts. And there are many symbols here, but all of them can be fixed by the experimental designer. The only thing we care about is the D, which is the diffusion coefficient. So we can accumulate the signal of the intensity that's decaying over time, and we fit the model to get the diffusion coefficient. This is done by collaboration with Dr. Amelia in our university, and she did the measurements for all of the colloidal system. And the conclusion we get is, for example, for the NaCl, they can only be used to change the diffusion outside the capsule, and there's no change for the water molecule inside, which means the salt cannot penetrate the membrane. We can utilize the same strategy to screen what kind of species can penetrate the membrane so that we can further utilize them as the cargo to deliver in other places. As I said, even the particle level transport is hard to study, and one effect is called the entropic bottling effect. When I was young, I was always fascinated by this simple magic trick called impossible bottle, where people put the large shape inside this bottle with a very narrow neck. I was confused how they managed to do so. During my PhD study, I figured out another phenomenon that's similar but also very interesting called jamming. Jamming means you have a smaller sheep inside the bottle, but if you have many of them inside, it's still very hard for them to exit because of kinetic jamming. This kind of thing gets more and more interesting if you relax these two constraints. That means you shrink the size of the sheep and you decrease the number of the sheep until the extreme condition, you only have one small sheep inside this bottle. We still have this kind of effect that prevents the sheep from exiting. This is called entropic bottling effect. It's purely from entropy, nothing to do with like energy. So this has been a very big topic at the beginning of 21st century. Many physicists did the simulation, but after which there's no follow-up because they cannot have experimental system to verify their results in these papers. So if you remember, our capsules is the identical structure to use in the simulation, which is the large cavity with the small hole. So we can measure the diffusion of particles by studying their transport. And surprisingly, we got a very big gap between their time scale. For small particles, they can exit within minutes, whereas for large particles, they need even months in order to exit, which is exactly what the entropic bottling effect predicts. However, due to the current experimental resolution, we are unable to verify the scaling law they got from this simulation, but we are working hard on that. So we hope to get more information in the future. However, we didn't achieve the original idea of impossible bottle, which is the very large object trapped inside this small container, because within a solid system, there's no way that you can push a hard sphere that's larger than a hole into the system. There's no way. But just like magicians are creative, soft matter scientists like us are also very creative. So we can use the soft interaction to artificially increase the size of the particle once they're inside. So essentially, we decrease the hole and increase the tracer, we get the impossible bottle trapped inside. This is called Wigner crystal, where the tracer particles start to feel the electrostatic repulsion from the wall, which is the container. If that's the case, particles will be separated far enough due to the electrostatics. And if they're trapped inside the container, like this spherical container, they need to self-organize themselves into the symmetric tracers, symmetric clusters, which you can see here. We can get all different kinds of symmetries as predicted in the molecular space. So this is the minimal energy configuration for all of the clusters. So no matter how fancy they look, they still fall under the passive transport regime, means everything can be equilibrated if you give them long time enough. But the living cell system always harness the energy of ATP to break the concentration equilibrium so that they can accumulate the things they like and to expel the waste they accumulated beforehand. So this is kind of non-equilibrium transport is very hard to realize in abiotic systems because you need to have an energy input that can be selectively consumed and transformed into this kind of cross-membrane motion. So in the final part, I'd like to talk about how we can utilize the power of active colloids to realize that. So we are going to use the group of active colloids called Phoretic colloids. Phoretic phenomenon, to put it simple, it's just the interfacial phenomenon between your colloidal particle and the surrounding fluid. If you zoom into the interface, usually like 20 nanometers, as long as you have a gradient present, it can be a temperature gradient or like a concentration gradient. This will form osmotic pressure, pushing a particle directional towards a certain way. So hematite particle, as I used to template the anisotropic hole before, they are not only magnetic and isotropic, they are also good catalysts to decompose the chemical field like hydrogen peroxide if you shine the blue light on top of them. When this is the case, you can shine the blue light to generate the gradient near this hematite cube so that they have the phoresis by themselves. And this phoretic phenomenon is very strong. They can be used to attract tracers towards them or expel them depending on the surface chemistry and the pH. Experimentally, we found that at basic condition, the tracers are usually attracted, whereas at acidic condition, they will be repelled. So we can use the global pH as the tuning knob in order to change the direction of the motion. So the Diffusiophoresis-Driven Transport is like a UFO manipulating objects using the tractor beam. It's very long range and remote. It's also like a white blood cell chasing a bacteria that they can sense the surrounding environment. So because of that, we can embed this hematite cube inside our microcapsule, and we can transform the passive cell into an active cell mimics. And we will use both the light and the pH as the tuning knob to achieve both the ingestion of the tracers and the expulsion of them. So in this video, we start from the ingestion, where it's conducted under basic solution. When you shine the light, the hematite starts to accumulate tracers from the outside. It's super effective. They can accumulate hundreds or thousands of tracers within a minute. And if you turn off the light, the tracers will start the Brownian motion. But once you click the light, they will start to be attracted again. When you finish the ingestion, you can simply lock them inside for a very long time due to the entropic bottling effect. And if you want to release them, you simply change the pH into acidic and do the same trick. And now all the tracers can be kicked out until the single cube is left inside. So just like a living cell, we accumulate different molecular species to do a synthesis. We can also accumulate different colloidal objects to do a colloidal synthesis inside. We can use them as the microreactor to create the hybrid colloidal particles that's otherwise very hard to make. So diffusiophoresis is not only effective, but also they are directional. That means we can also use the same mechanism to attract anisotropic particles like rod-like particles. On the left video, we are using the same strategy to attract a silica rod, which has the length bigger than the hole. So the only way for them to enter is to align the perpendicular towards the hole so that they can slide in and be accumulated. As you can see in the video, they show the same effectiveness. They can accumulate hundreds or thousands of rods inside within minutes, which is highly effective compared with other mechanisms we can achieve. And this kind of phoresis is a surface phenomenon, so it's very hard to predict whether they work for the complex surface. So we tested our system with the biosurface like E. coli, which is both anisotropic and complex in terms of the surface molecules and structures. And surprisingly, the same mechanism works, and they can accumulate even large E. coli into the system, which paves the way for us to utilize them to study biological delivery or accumulation of biohazard. If you zoom in on the video, this is the hole, and the E. coli will slip from the hole into the capsule. This is the second entry. So just to give an outlook for the future, living organisms have different structures and different shapes. So our colloidal microcapsule or colloidal cell mimics is just the starting point. In the future, we want to study more structural diversity for these microcapsules. And also, living cell has many internal pathways. That's the interaction of the machines inside the cells. We can put inside a variety of different active particles to study their interaction as well, such as the mimic for this internal pathway. Living organisms also have the multicellular communication, which means many different cells will organize together, either packing or swimming. So we can put inside many active protocells and to study their interaction as well. Maybe they will give us more surprising functions. And finally, as I start in the beginning, TPM can be used to make many different shapes and functions. It never disappoints us. I have no doubt that TPM in the future will give us more differentiated protocells, and we can use them to produce more varieties of functions. Quick summary. So I start from talking about the scalable bottom-up synthesis of colloidal microcapsules using the self-inflation method, and I get complete tunability of the parameters, including the size, entrance, and the membranes. Finally, I achieve both the passive transport and active transports using this microcapsule. So I'd like to thank my supervisor, Professor Stefano Sacanna on the left, together with our group members here. And also on the right is our collaborator. On top is Professor Zhe Xu at NYU. And on the bottom is Professor William Irwin from University of Chicago, who is the expert in the fluid dynamics. And finally, if you're interested in any kind of particle I just mentioned, you can check our YouTube videos. We have all the tutorials ready for you. And also, if you don't have a chemical lab, no worry. We also have a library called COL where we can send you some particles to develop a collaboration. And if you want to make some capsules and face difficulties, you can also send me the email and I can show you more tricks within that recipe. With that said, I'd like to end here and to take some questions.
Okay, thank you, Zhe. Thank you for a wonderful talk. Very interesting work. So let's see if we have questions or comments from the audience.
Oops.